Hello everyone, welcome all of you in my new video on my channel. Today I am going to discuss the biochemistry paper of West Bengal University of Health Sciences. Right, so let's start. So the first question is methotrexate, a widely used anti-cancer drug, acts by inhibiting. So methotrexate is basically a chemotherapeutic agent which is used to treat the cancer. So as DNA are synthesized from the folic acid, right? Folic acid converted into DHFA, then it will be getting converted into THFA, and then by the lots of sequence of reaction, it is going to get converted into the DNA, right? So here one enzyme is there in the pathway, that is dihydrofolate reductase, which converts DHFA to THFA. So methotrexate acts basically on this enzyme, that is on the DHFA reductase, so that the formation of DNA or the formation of genetic material stops, right? And if there is no genetic material, then no further cell division is going to happen, and it helps in halting the process of cancer, as the cancer is the uncontrolled cell growth, right? So the answer is dihydrofolate reductase. Next, a 1-year-old child brought to the pediatric OPD. Mother complained that the child has a mousy odor in the urine. Answer is quite evident from this, that is, it is a case of phenylketonuria, as mousy odor urine is seen in cases of the phenylketonuria. On examination, it was seen that the child has many developmental delayed milestones and the hypopigmentations of hair and skin. Ferric chloride test and the Guthrie's test was positive. Which enzyme deficiency can lead to this disorder? So there are many clinching points given in this question. As you know, this one is the first, that is the mousy odor urine, right? Mousy odor urine is a clinching point. Second is, that is the hypopigmentations of hair and skin, and the last is the ferric chloride and the Guthrie's test. All are the easy given clinching points, right? All are found in the phenylketonuria, where enzyme phenylalanine hydroxylase is deficient, and if there is phenyl hydroxylase deficient, then phenylalanine is not going to convert into tyrosine, and if there is no tyrosine, then no melanin synthesis is there, so there is whitening of the hair and the skin, right? So the answer here is phenylalanine hydroxylase, and mousy odor urine is due to the phenyl acetate, right? Next question. Enzymes of the glycogen metabolisms are under the allosteric regulation. Identify from below which of the following is not an allosteric regulator of the same. So what are the allosteric regulators? Allosteric regulators are those compounds or those substances which acts on the enzyme other than the active site binding, right? So by binding to the enzyme, it modulates the action of the enzymes, either activated or inactivated. So here in the liver glycogen metabolism, you have allosteric regulators like glucose 6-phosphate, ATP, glucose, AMP in the muscle. So here the answer would be insulin. Insulin is not an allosteric regulator. Rest all are the allosteric regulator of the enzyme, either glycogen phosphorylase or glycogen synthase. It is not mentioning about one enzyme. So in glycogen pathway, you have both, that is breakdown and synthesis of the glycogen. So here you have to consider the activator or inhibitor of the both enzymes, that is glycogen phosphorylase as well as glycogen synthase, right? So here the answer would be insulin. Next question. In product of one cycle of beta oxidation of odd chain fatty acid, as you know, even chain fatty acid after beta oxidation gives two molecules of the acetyl CoA, and the end product of the odd chain fatty acids are the acetyl CoA plus propanoyl CoA. So the here answer would be acetyl CoA plus propanoyl CoA. Next question. Which one of the following has a pseudouridine in the structure? So as you, maximum number of modified bases are seen in the transfer RNA. As you can see, transfer RNA has 5 arms. One arm is the dihydrouridine arm, right? This is the dihydrouridine arm. Other than that, you have the T arm. T arm is which contains the ribothiamidine. One is a acceptor arm and one is the anticodone arm. So pseudouridine is basically seen in the all the modified bases, maximally seen in the transfer RNA, right? Next question. In hospitals, rice tube is discarded in. All the rubber tubings, syringes, IV bottles, these all are discarded in the red bag. So the rice tube will be also discarded into the red bag. You have, other than that, yellow bag, blue bag, black bags. So in the red bags, basically we discard plastic waste such as catheters or the rubber wastes. Catheters, injections, syringes, tubings, and the IV bottles, right? So here answer would be red bag. Now the next is the precursor of synthesis of polyamines. As the name suggesting, polyamines means compounds containing more than one amino group, such as polyamines are putrescine, asparmine. These are the compounds of polyamines, right? And these are synthesized from the ornithine and S-adenosylmethionine, right? So here answer would be ornithine and S-adenosylmethionine start the process by which polyamines are synthesized in the body, right? Enzymes associated with the reverse cholesterol transport is, so what is reverse cholesterol transport? So reverse cholesterol transport is basically transporting the cholesterol from the peripheral tissues to the liver, right? So reverse cholesterol transport basically means transporting of cholesterol from the body to the liver, right? And in this helps an enzyme that is known as LCAT, right? LCAT, what is the function of this LCAT? LCAT esterifies the cholesterol and it is associated with the HDL. So LCAT is associated with the reverse cholesterol transport system. What is LCAT full form? Lecithin cholesterol acyl transferase, right? Okay. Next question. After being synthesized in the radiculoendothelial cells, bilirubin is transported to the liver through which of the following transporter? It is very easy. After the formation of bilirubin, it gets combined with the albumin and then it is transported in the blood. So your answer would be albumin. Right. Now the next question. A 2-year-old child presented with the liver enlargement. On investigation, following features are found. In this question, I think this question is incomplete because liver enlargement is seen in almost all 4 conditions. Right. So I think question is wrongly framed. If you have to give some sign and symptom in this question, only then we have marked it, right? So this question is something wrongly framed. So but that in all the conditions, whether it is von Gates disease, in galactosemia, in fructose deficiency, or in the G6PD deficiency, you have hepatomegaly, right? So all the conditions having the hepatomegaly. Thank you.